Good morning, students. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, Hyderabad, is here to talk on IMViC tests. IMViC series or tests are a group of 4 individual tests that are commonly used in identifying bacterial species belonging to the family Enterobacteriaceae. It is used or designed especially to differentiate gram-negative intestinal bacteria, particularly Escherichia coli and Enterobacter Klebsiella groups, on the basis of their biochemical properties and enzymatic reactions in presence of specific substrates. This particular test is used to identify coliforms. Coliforms are gram-negative aerobic or anaerobic rods which produce gas from lactose within 48 hours. The presence of some coliforms indicates fecal contamination. Each of the letter in IMViC stands for one type of test. I for Indole Test, M for Methyl Red Test, V for Voges-Proskes test, and C for Citrate Utilization test. The small I or the lowercase I is added for the ease of pronunciation. IMViC is an acronym that stands for 4 different tests. Let's begin with the first test, Indole Test, which stands for I in IMViC. The aim is to determine the ability of a microbe to degrade the amino acid tryptophan. Principle: tryptophan, an essential amino acid, is oxidized by some bacteria by the enzyme tryptophanase, resulting in the formation of indole, pyruvic acid, and amino acid. The indole test is performed by inoculating a bacterium into tryptone broth or a peptone broth, where the indole produced during the reaction is detected by adding a reagent known as Kovacs reagent, or chemically it is known as para-dimethylaminobenzoaldehyde, which produces a cherry red reagent layer. So the reactions involved in this particular test are: tryptophan is degraded into indole, pyruvic acid, and amino acids by the enzyme produced by the microbes. The enzyme produced is tryptophanase. The indole produced in this reaction is detected by para-dimethyl para-dimethylaminobenzoaldehyde. This particular reaction undergoes in presence of HCl and alcohol, and it results in dehydration, removal of one water molecule, and production of quinidinol red-violet compound. Interpretation: development of cherry red color at the interface of the reagent and the broth within seconds after addition of Kovacs reagent indicates the presence of the indole, and the test is pronounced as positive. If no color change is observed, then the test is negative, and so the organisms are not capable of producing any tryptophanase enzyme. E. coli is indole positive while Enterobacter Aerogenes is indole negative. Precautions to be taken during this particular experiment is: take a fresh tryptone broth for best results. Repeated gentle shaking of tubes is necessary for 10 to 15 minutes after addition of Kovacs reagent for the development of deep red color. Third precaution is: do not incubate the inoculated tryptone broth tubes more than 48 hours, because after 48 hours of incubation, indole itself may be attacked and further degraded. If this happens, the indole will eventually disappear and you may not get a misleading, you may get a misleading negative test result. The picture of Indole test. Tube on the left is positive with a red ring inoculated with E. coli. Tube on the right is negative inoculated by Enterobacter aerogenes. The next test or the second test in IMViC is Methyl Red Test. In short, it is known as MR test. Aim to determine E. coli and Enterobacter aerogenes, the ability of microbes to oxidize glucose with production and stabilization of high content of acid end product. The methyl red test is used to differentiate two major groups of facultative anaerobic enteric bacteria that produce large amount of acid and those that produce neutral product acetoin as end products. Both these are performed simultaneously. MR test is simultaneously performed with Voges-Proskus test. Usually, we obtain opposite results for MR and Voges-Proskus test. In these tests, if an organism produces large amount of organic acids like formic acid, acetic acid, lactic acid and succinic acid from glucose, the medium will remain red, a positive test on addition of methyl red, a pH indicator. The pH indicator value of methyl red is 4.4. In other organisms, methyl red will turn yellow due to the elevation of pH above 6 because the enzymatic conversion of the organic acids to non-acidic end products such as ethanol and acetoin. We can see glucose produces lactic acid, acetic acid and formic acid, and when methyl red indicator is added, it turns into red color due to production of carbon dioxide and water. Whereas in E aerogenes, glucose produces acetic acid and later immediately it will produce 2,3 butanidol plus CO2 plus water, and on addition of methyl red it turns into yellow color where the pH is 6. So the indicator methyl red is red in at pH 4 and yellow at pH 6. Picture of methyl red test. Two tubes. On the left is positive inoculated with E. coli and on the right we can see a negative inoculated with Enterobacter. Carbinol or acetoin from the organic acids that results from glucose metabolism. This test is characteristic for Enterobacter aerogenes. Test identifies bacteria that ferment glucose and lead to 2,3-butanidol accumulation in the medium. Glucose plus half oxygen produces 2-pyruvate and liberates CO2, in turn produces alpha acetoacetate. Alpha acetoacetate gets converted into acetoin by decarboxylation. Acetoin by decarboxylation in turn produces 2,3-butanidol. Acetoin reacts with alpha naphthol. Alpha naphthol is the reagent which is added in this particular process after incubation for 48 hours. So the acetone produced by the microorganism reacts with alpha naphthol in presence of 40% KOH and absolute alcohol to produce diacetyl plus creatine. Diacetyl results in producing a pink colored complex. So in this experiment, the result or interpretation is development of a crimson red color indicates a positive test for Enterobacter aerogenes, and no color change indicates negative test, which is observed in case of E. coli. Picture of Vergus-Proskus test. On to the left, we can see a positive MR test. Onto the left, we can see a positive VP test, which is positive for Enterobacter aerogenes. Onto the right, we can see a negative test for E. coli, where there is no color change at all. The fourth test in this particular experimental is citrate utilization test. Citrate test is used to determine among enteric bacteria on the basis of their ability to utilize or ferment citrate as a sole carbon source. The utilization of citrate depends on the presence of an enzyme, citrase, produced by the organism that breaks down the citrate to oxaloacetic acid and acetic acid. These products are later converted into pyruvic acid and carbon dioxide enzymatically. So the aim of this particular experiment is to determine the ability of the microbe to ferment citrate as sole carbon source. Principle: citrate a sole carbon source for their energy needs. Presence of a citrate permease that facilitates transport of citrate into the bacterium. Sodium citrate as the carbon source and ammonia as nitrogen source. pH indicator used in this particular experimental is bromothymol blue. This test is done on a slant since oxygen is necessary for citrate utilization. When bacteria oxidize citrate, they remove it from the medium and liberate CO2. CO2 combines with sodium supplied by sodium citrate and water to form sodium carbonate, an alkaline product. This raises the pH and turns the pH indicator bromothymol blue to blue color and represents a positive citrate test. Absence of a color change is a negative test. Citrate negative cultures will also show no growth in the medium and the medium remains green. The reaction here is sodium citrate in presence of citrate permease and citrase produce pyruvic acid, oxaloacetic acid and CO2. Excess sodium from sodium citrate reacts with CO2 and water to form Na2CO3, which reduces the pH level, leading to conversion of color green to blue of the medium in presence of a pH indicator known as bromothymol blue. Picture or citrate utilization test. Negative tubes are green in color and do not bear any growth of the microorganism, whereas positive is blue in color with growth of the microorganism. Let us compare the IMViC results between Escherichia coli and Enterobacter. Escherichia coli exhibits indole positive, methyl red test positive, Vergus-Proskus test positive, and citrate utilization test negative. The first tube Indole test is showing the indole ring. Second tube is MR test, which has turned to red color. Third tube is IMVP test where there is no color change, and the fourth tube is citrate utilization test where the tubes are in green color. Enterobacter Aerogenes. Indole test is negative, Methyl Red test negative, Vogus Proscus test positive, citrate utilization test also positive. The first tube is Indole test where we cannot see any red, cherry red color ring, negative. Second test is methyl red, the tube is in yellow color. VP test, again we can see a pink color indicating positive test. Citrate utilization test, the tube has turned into blue color. Thank you.